Alright guys, so this lesson is going to be our last lesson in this unit. We're only going to take a look at prokaryotes and viruses and just kind of that intro about microbiology for this first test as we start to go through all of the living things. And so you've done the prokaryotes and we've taken a close look at the cells that are in domain bacteria and domain archaea. And as we're studying microbiology, there are many other things that could be studied within that umbrella. So protists are going to fit under microbiology, or many protists will, some fungi will. But another thing that will be covered under microbiology are viruses. Now, I do want to make clear, we'll talk about this here in a second, viruses are not living, but because they do affect living cells, and as we can see, nearly all life forms are going to have viruses that can infect them, they're definitely going to fall somewhere in biological study. And so they kind of fit in with these microorganisms. So even though they're not an organism, we study them kind of along with bacteria and other pathogens. Now, viruses are really kind of these biological weirdos because they have some features of a living cell. They can harm living cells, but they are not living cells themselves. And so we're going to see that they vary in structure. So there's going to be different ways that viruses can be built. They vary in how they replicate, and they vary in the host that they target. Now we're going to see this multiple times, but viruses are always specific. It's not like there's one virus that goes around and infects all the other organisms. Viruses are specific to species, or they're specific to groups, or they're even just specific to certain cells within an organism. Now, in terms of where did they come from, honestly, a lot of the origins are unknown. We don't know exactly when they came about, but essentially they're what we call obligate parasites, and that they take advantage of living cells, and they're going to use them to reproduce. So as I mentioned, they are not considered living organisms. So while we may study them in microbiology, we would not consider them microorganisms. They're not considered living for many reasons. Way back in the very beginning of biology, we looked at the characteristics of life. I think it was the very first lesson that we did, and we took a look at what pretty much constitutes a living cell or a living organism. And there's kind of a list of criteria that make up a living thing, or that you will find occurring in living cells. And so one of the big ones is that they are not cells. Okay, we're going to see in a minute that this is acellular is another way to say that. But they don't have the cellular structure. They don't have a cell membrane. They don't have cytoplasm. They don't have ribosomes to make proteins. They don't have the components that both your prokaryotes and your eukaryotes have. Now, while they can reproduce, they cannot do it on their own. So they require a living cell in order to reproduce. And that's what makes them those obligate parasites. They're going to take advantage of a host. And then some other things that they do not do that is going to knock them out of that living category is they don't grow and develop over time. You don't see viruses maturing, or you don't see viruses increasing in size. They don't respond to stimuli like light or food. They don't even need food. They don't metabolize. They don't use energy. They don't make energy. They don't have to have light, or they don't have to have food, or they don't have to have ATP. They essentially just take over a host cell and use all of the host cell's machinery just to make new viruses. And then they also don't maintain homeostasis. They don't have that internal constant condition. So in terms of the characteristics of life, the only things that they do kind of share is they do have genetic material. Now, one difference is this word "or," okay, where cells will have both DNA and RNA, a virus will have DNA or RNA. And then because they have genetic material, they can change over time. This was one of our characteristics of life. But as you can see, all of these other characteristics they do not meet. So we do not consider them actual microorganisms, even though we may study them in microbiology. Now, how do we kind of look at their structure? How do we compare them to a living cell? Well, one difference is they're way smaller. Back when we covered cells, they were usually measuring anywhere from 2 to 15 micrometers. In fact, viruses are so small we could measure them in nanometers, which is billions of meters. So they're quite a bit smaller than even the tiniest bacteria. And as I mentioned earlier, they are acellular. When you put an A or an "an" in front of something, it means not. So they are not cells, they are not cellular. And so if they're not cells, then what are they? Well, essentially viruses are two things. They are some kind of nucleic acid. It could be DNA or, there's that important word, DNA or RNA. So there's some type of nucleic acid in a protein coat, and we call that protein coat a capsid. So those are the two components that all viruses share. Now there's going to be some different variations of that, but notice you don't have a phospholipid bilayer. You don't have a cell membrane that's regulating what's in and out. You don't have a cell wall. We aren't even using the term cell in their structure. They're essentially some kind of genetic material surrounded by protein. Now, you might find some viruses that have an envelope, and what they'll do is they'll actually steal some of those phospholipids and proteins from the host, and they'll create this additional layer outside of their capsid, but it's not the same structure as a cell membrane, and it doesn't do the same thing as a cell membrane. And they may also have their own enzymes, and they may have some other proteins, but they're lacking most of the machinery needed to replicate and build new viruses. Now, in terms of their protein capsid, that's going to kind of determine their shape or whether or not they have an envelope, things like that. And so we can, for the most part, and there could be some exceptions here, we can put them into four main groups. They could be helical, and normally the helical are kind of long, and they're filamentous. They look like long filaments. One example of that is the tobacco mosaic virus. This is actually a plant virus, and it was the very first virus to be discovered. They could be polyhedral, and so poly means many. These are multi-sided geometric type shapes. A lot of them are what we call icosahedral, which means they have 20 sides to them, like this adenovirus here. They could be spherical. A lot of viruses that infect humans, like the flu or HIV or the common cold, a lot of those are these spherical, and they have envelopes around them. Or they could be a complex, which for the most part of these bacteriophages. Now this is a word that I don't know if we've seen it before yet this year, but sometimes this is just shortened to a phage. But bacteriophages are actually viruses that infect bacteria, and they normally have this little head and tail type structure. So you can see that they can come in different shapes, and all that's really different is that protein coat and whether or not they have those additional layers like an envelope. Now when it comes to the genome, we've seen this word before. We saw this with prokaryotes, so just a reminder. The genome refers to the genetic material, the DNA, all of the genes that you will find in an organism. And so the genomes are very small because they don't have to do all those life processes. They really don't have to do a whole lot other than make some new proteins. They don't have to do chemical reactions like cellular respiration, and so they don't need a lot of enzymes and things like that. And so they really only need genes to build the proteins that the virus can't steal from the host. So they don't have really long strands of genetic material. Now, as we mentioned earlier, their genome could be a DNA genome or it could be RNA. They don't usually have both in one. Whether it's DNA or RNA, and this is very different from living things because in a living cell, DNA is always double-stranded. But in a virus, it could be a single-stranded DNA, it could be a double-stranded DNA, it could be a single-stranded RNA. You know how when we talked about chromosomes, we said that eukaryotic chromosomes are linear and prokaryotic chromosomes are circular? Well, when it comes to your viruses, they could be either one. So it kind of varies. It just depends on the individual virus. Now, something they do have in common with living things is because they have that genetic material, they can evolve. So just like when we're copying our DNA, we can get random mutations, or if we're making RNA from our DNA in transcription, we could make mistakes. Those same things can happen with the genetic material of a virus. So just like our cells can evolve as mutations occur, viruses can also evolve. And that actually makes them kind of tricky because if we try to identify viruses or treat viruses in certain ways or create vaccines for viruses, what can happen is if they mutate, then those methods that we've developed may not be effective anymore. And you guys see this in your daily lives, like there's different strains of the flu. And when you get a flu vaccine, you just have to hope that it's going to be the strain that's kind of most common that year. It doesn't always guarantee that you won't get the flu because sometimes there's a different strain around. Something like HIV, it mutates pretty rapidly, and so as they develop medications to help treat it, they have to change because the virus changes. And so this can be a little tricky for us when it comes to studying them and trying to get rid of them or trying to stop them from hurting our cells. Now a little bit more about the genomes. Now, depending on whether it's a DNA or RNA virus, if it's a DNA virus, normally what they're going to do is they're going to tell the host cell to copy that viral DNA, and then they're going to transcribe, which means they turn it into RNA, and then translate it to make the viral proteins needed to build new viruses. Now, it really there can be some variation there, but in general, we go straight from DNA, we copy it, we turn it into RNA, and then we make proteins, kind of like what we studied back in first semester. Now, if it's an RNA virus, many of these have a special enzyme called reverse transcriptase, and it's the opposite of transcription. So when we covered protein synthesis, we learned that transcription was DNA to RNA, and so what this enzyme does is it takes the viral RNA and it copies it and turns it into DNA first, and then it will go about making copies and then transcribing and translating. Now, for some reason, in these retroviruses, which is what we call them, the mutations usually occur more frequently. So a lot of times these are harder to track down and treat, and because they're constantly changing. And HIV is an example. This is a really tricky disease, tricky virus, because it mutates pretty frequently. All right, so host specificity. So in order for a virus to reproduce, it's going to have to infect a host cell, a living host cell, and so it's always going to be specific. And when we talk about specificity, pretty much these guys, your proteins, are always going to be involved. Proteins always have a very specific shape and they always do a very specific function. Now, a virus can only infect a cell if the proteins on the virus can attach to proteins on the host. Okay? Now, something I want you to understand is that our cells, our living cells, naturally have these protein receptors. Okay? We have protein receptors for things like hormones. We have protein receptors for things like neurotransmitters. We have protein receptors that involve things like cell transport. Now, it would be really dumb for our cells to make proteins specifically for viruses, and we don't do that. We don't make proteins for the virus. But what happens is we have these protein receptors that are normally on our cells, and they actually have a different function, but viruses have evolved a way to utilize them. So they found a way to attach to those receptors, and then normally when something binds to a protein, it changes shape. And so if that virus can attach to the receptors, it will a lot of times allow entry of that viral genome into our cells. So viruses are tricky little things, and they've kind of evolved and figured out ways to utilize these host cell proteins. So once again, we don't have them for the virus. The viruses are just able to use these receptors that we already have. But it's always specific. So some viruses have a very broad host range. They can affect many organisms, many different species. Like rabies, for instance, can, for the most part, infect mammals. And then some viruses are super specific, like HIV. It specifically targets a type of helper T cell in our immune system. And, it doesn't affect any other cells in our body except for these particular helper T cells. And that's why they affect our immunity and they affect our ability to fight disease. So the specificity just has to do with proteins, and some are more specific than others. But like a bacteriophage that infects bacteria would not be able to harm an animal cell because it can't attach to those proteins. Now, when it comes to a viral infection, in general, there's kind of 4 major steps. So the first thing that has to happen is the virus has to be able to attach using those proteins. Once it's attached, it has to find a way to get the genetic material into the host cell. Now, depending on the virus, there's different ways that this can happen. It could just kind of inject the genetic material into the host, and so it kind of enters in what we call naked. It doesn't have that capsid around it. Sometimes it'll fuse with the envelope, or sometimes the cell will kind of pull it in by endocytosis. But no matter what, the genome of the virus has got to enter the host cell. Now, once that genome is in there, that virus is going to use the host to replicate their genetic material and build the proteins needed to make the new viral capsids. And so essentially they're going to build the new viruses, and then they're going to find a way to get those viruses out of the host. Now, once again, sometimes this could be the viruses will lyse the cell or cause it to bust open and kill the cell, or sometimes they just kind of bud out in what kind of looks like exocytosis. But either way, the new viruses, once they're made, they have to get out of the cell, and then they go on and try to infect nearby cells around them. So here's kind of the basic process. So here's the flu virus. You can see that it's going to attach, then it's going to trick our cells into bringing it in, kind of in a membrane, and then it gets broken down so that genetic material is exposed, and then we copy the genetic material and build the new viruses, and then they leave our cells by budding. But ultimately it's going to damage the cell in some form or fashion. And so this is why when we have a virus, we feel bad. Either A, it's damaging our cells, B, we're having an immune response, and we have all different ways that our immune system responds, and normally it's C, both of those. So the reason you feel crummy when you have a virus is your cells are being damaged and your immune system's kicking in trying to get rid of it. And so we have symptoms like maybe a snotty nose, or you may have vomiting or diarrhea or things like that. So part of it's our body trying to get rid of it, and part of it's our cells literally being damaged. So they could cause our cells to die, either through lysing our cells, making them bust open, or they could cause the cell to trigger apoptosis, which is that cell death. The cell kind of breaks itself down. And then sometimes they don't kill the cell, but they damage the cell enough that it doesn't work properly. And these can be a little dangerous because they can be what we call oncogenic viruses. The prefix onco refers to cancer, like an oncologist or oncology. So oncogenic viruses, what they do is they damage the cell and they affect its ability to go through the cell cycle. Some common examples of this is HPV. A lot of times now people are maybe starting around 10 or so. They encourage like young people to get the HPV vaccine. That's the one that can cause like genital warts. It does lead to cervical cancer in females, but it can also lead to things like oral cancer in males. Hepatitis virus could potentially lead to liver cancer, and then some leukemias and lymphomas could be the result of viral infections. So either way, the cell is going to be harmed. If it's infected by a virus and the virus uses it to make copies of itself, it's either going to kill the cell or damage the cell in some way. And so as I mentioned earlier, the reason we have disease or the reason we have all these symptoms is it's because of our cell damage and because our immune system is kicking in trying to get rid of it. Now, in bacteriophages specifically, so these are the viruses that infect bacteria, there are 2 types of infection cycles. We have lytic, and I want you to notice that this is the shorter word, and this one is going to be very quick and immediate, okay? Or we have lysogenic, which is the longer word, and this one is going to take a while, okay? So in the lytic cycle, what happens is the bacteriophage attaches, it injects its genetic material, and it immediately takes over the host. So as soon as it injects that DNA or RNA, it starts using that host to make copies and make new proteins, and within a very short period of time, it's going to build new viruses, and it's going to lyse that cell open, which is where you get that term lytic. So the key to lytic cycle is it's an immediate takeover. Now, a little trickier than that is the lysogenic cycle. And so what these bacteriophages will do is they inject the genetic material, but they don't immediately take over the cell. Instead, the viral DNA or the RNA becomes part of the host cell genome. And so we're talking bacteria here, and they can reproduce very quickly. So now every time that bacteria copies its DNA, it's copying that viral DNA with it. And so what can happen is one cell can get infected, and then it just hides in that cell. Well, when that cell copies itself, now you have 2 bacteria that have the virus. And then those cells copy, and now you have 4 bacteria that have the virus. So what it does is it just kind of hides. It's not really hurting the cell. This is what we call latency. And some of our animal viruses do this too. They kind of hide in the genome for a while. Herpes is an example. HIV is an example. You know, just because someone looks clean or just because someone looks like they're healthy doesn't mean they aren't harboring a virus. Eventually what will happen though is, usually due to environmental triggers, it'll cause that viral genome to kind of exit the host genome, and then it'll start going through the lytic cycle. So usually due to some kind of environmental change, it'll trigger those hiding cells to start doing the lytic cycle. And so at that point, we have many bacteria that are infected, and now they're all going to start making new viruses, and they're all going to get lysed. So once again, the lytic cycle over here is immediate takeover. So pretty much it injects the DNA, it takes over the cell, and it kills the cell. Now over here in the lysogenic, notice that that viral DNA gets incorporated into the host cell. The cells will copy themselves, making multiple copies of the virus, okay, and that can happen for generations, and then eventually those cells are going to go through the lytic cycle, and they're going to cause that cell to die. Now, some common viruses that you may see in humans, I'm sure some of you guys have had some of these, like the flu, the common cold. This is mono, if you've ever had mono. Coronavirus, obviously that was kind of a big deal a few years ago, and it's still hanging around now. West Nile, that's transferred by mosquitoes. Chickenpox. Warts. This is the human papillomavirus. This is both genital warts as well as like normal warts that you could get like on your fingers or toes or somewhere like that. Hepatitis, rabies, adenovirus can cause things like pink eye or just respiratory infections. So there's all different types of viruses. Now there's a couple other little strange weirdos that we're going to talk about. Now they're acellular as well, which means they're not cells, but they're also not classified as a virus. So remember that viruses have 2 components. Viruses either have DNA surrounded by protein or RNA surrounded by protein. They have these 2 main components of genetic material and protein coats, okay? Well, these dudes don't even meet the virus criteria. So a viroid is just a little piece of RNA. It doesn't have a capsid. And these specifically infect plants. We don't really have any issues with these viroids, but if you were trying to grow crops and they got some plant viroids, that would affect your crop. And then prions, these can affect humans, but prions are abnormal proteins. And what they do is they're normally in the brain, and these abnormal proteins will trigger nearby proteins to become abnormal. And so they're degenerative nervous system diseases. And a couple examples, there's mad cow disease, which can transfer to humans if you eat the infected meat, or kuru, which is kind of a human version. This was common in the island of New Guinea, where people were practicing cannibalism. And so prions, essentially, you can transfer it by eating like this infected meat, and then it could cause proteins to become abnormal in the brain. And that's what makes, people, they're going to act different if you're damaging the brain tissue. So once again, these are not viruses, because this is only genetic material and this is only protein. So they're just some other little infectious weirdos. Now, we didn't really talk about these virusoids. I haven't seen much about these personally, but the viruses have both genetic material and a protein coat. Viroids are just genetic material, and prions are just proteins. All right, and the last thing we're going to talk about in this lesson is just some general information about disease. So we're kind of still in this microbiology unit, and we're just focusing specifically on prokaryotes and viruses right now. But remember that all different types of organisms or things could be pathogens. A pathogen, by definition, is just something that causes disease. So we know that bacteria can cause disease. You looked up some of those in your lab the other day. We know that viruses can cause disease. You're going to do that in your activity tomorrow. There are fungal diseases like athlete's foot or yeast infections. There's protist infections like malaria. And so we're going to take a look at those when we get to protist and fungi. But when it comes to these microbes, if they are pathogens, they could cause disease in different ways. So they could literally break your cells down for food. They could be parasites where they're using you for nutrition, or they could be using your cells for reproduction like the viruses do. They could literally just destroy the cells, or they could cause those cells to become damaged and disrupt the workings of the cell, or these pathogens could release toxins that damage our cells. So either way, our cells are getting hurt, but there's different ways that could happen. And then the last thing I want to do is kind of compare bacterial diseases with viral diseases, because there are some key differences between the two. Bacterial diseases are caused by living cells, and viral diseases are caused by viruses, which are not living. Now, prevention can be similar, okay, like there are vaccines for both. And what a vaccine is you expose your body to the pathogen so that you kind of get a weakened or a dead version. And when your body gets exposed, it will build up immunity to it. So it doesn't mean you'll never get the virus, but if you do, you should not have nearly as severe of symptoms, and you should be able to fight it faster, okay? Now, sometimes the vaccines can kind of help prevent you from getting it, but sometimes they just lessen the severity of the symptoms if you do get it. Either way, vaccinations can be used for bacterial diseases as well as viral diseases. And of course, these bacterial cells are living, so we could heat them and kill them. We could use disinfectants like alcohols. One of the best things to do with any kind of illness is just to avoid the sick people. Hand washing, try to stay away from, the coughs and the sneezes and the germs and things like that. Food preparation is really important. You need to cook the food properly. You need to store the food properly. Make sure that, if you're dealing with meat, you're cleaning your utensils and things like that, because some of those bacterial diseases like salmonella, you just want to make sure your areas are clean and we're cooking the chicken all the way and things like that. Now, when it comes to treatment for bacterial diseases, we typically use antibiotics, and antibiotics are specific to bacteria. Okay, so they're going to block the growth and reproduction, and usually if you have a bacterial infection, for the most part, you can get on some antibiotics and it'll knock it out pretty quickly. Now, with viral diseases, it is a little bit different, because remember, they are not alive, and so you can't really kill something that wasn't living to begin with. So really, your best thing with viruses is prevention, so we can vaccinate for viruses. Also, once again, avoiding sick people, washing your hands, and just kind of, be protective. In terms of treatment, normally you can just treat the symptoms. Most of the time, if you have a virus like the common cold, it just has to run its course. And if you go to the doctor, they're going to say go home, get lots of rest, drink lots of fluids, because your best defense against viruses is your own body. It's your immune system. And so you have to take care of yourself so that your immune system can fight it off and build up that immunity. Now, we can take medications like cough syrups or some Tylenol for fever, but all we're doing is treating the symptoms. Those types of medications are not treating the virus directly. It's just helping you feel better as your body fights the virus. Now, there are some antiviral medications that exist. They don't really cure a virus. It's not like an antibiotic where you can go in and kill the bacteria, but they could try to slow down the replication of viruses or prevent protein production of the viruses. But like I said, they're just kind of a cure, and they kind of help maybe alleviate some of the symptoms. So once again, no antibiotics for viruses. If you have the flu, you can't go get some amoxicillin, okay? So viruses is more about treating the symptoms and letting your body do the work, letting your immune system fight it, whereas with bacteria, you can take those antibiotics. And so just a little more information to kind of finish up. Vaccinations can be used for both, kind of being clean and good hygiene and proper food, and being healthy and eating well and seeing your doctor. All of those things can help prevent both bacterial and viral infections. And then once again, antibiotics would only be good if it were a bacterial infection. So, all right, that's going to wrap up our unit, or our first unit in microbiology, with our prokaryotes and our viruses. You're going to do some viral practice over the next day or so, and then you'll be gearing up for your test here in a few days. So if you guys have any questions or concerns, feel free to reach out. Otherwise, I hope to see you soon.